The next topic for Chapter 8 will be on genetic mutations. So we talked about those horizontal gene transfer methods, transformation, conjugation, transduction. Those are methods by which bacterial cells specifically can change genetically through transfer of DNA from a donor cell to a recipient cell. There are different ways that can happen. Now we're going to talk about another way that organisms can change genetically, and that is through mutations in the genetic code. All right, so we've talked a lot about going from DNA to RNA to protein, and we talked about the codon table and how a codon, a series of 3 bases on a messenger RNA, will specify an amino acid that's going to be included in the protein that's being built. So what is a mutation? Your textbook has some good discussion of different types of mutations and the definitions and so forth. And we're going to have a lab activity. It's not going to be one of the Learn Smart labs. It's going to be something that I'm going to assign separately from that. That'll have you go through on paper and actually make mutations and see the impacts of those. And I encourage you to take that genetics activity seriously once you were working on it, because my experience has been using that activity in class that it really helps students understand genetics better when they actually have to sit down and kind of work through a few problems on it. All right, so a mutation, again, is going to be any change in the nucleotide base sequence of a gene. And so we have an example here. This is going to be a pretend gene on DNA. And so in red right here, this is the sequence of our pretend gene. And in red, we're seeing this is the DNA version of the gene. We're only seeing one strand. Remember, real DNA has 2 strands. But this is our the template strand on the DNA. And then here we see the messenger RNA. So an RNA polymerase enzyme does transcription, hopped onto the DNA, read this template, manufactured the messenger RNA, and here's the messenger RNA. So we've got AUG, ACC, etc. Then this messenger RNA here in purple goes off to a ribosome, translation takes place, and we end up with our peptide. Remember, peptide is another term for a short chain of amino acids, just a few amino acids linked together. And so when you do translation on this, there's your start codon AUG. AUG codes for methionine. ACC is our next codon. That codes for an amino acid called threonine. CAC codes for an amino acid called histidine. And you keep going, and then finally you'd hit UGA over here. That's a stop codon. So there's no amino acid specified by UGA, so your peptide translation is finished. We've made our peptide. All right, and so here we see an example of a missense mutation. And a missense mutation is a type of point mutation. So we've got some new terminology here. A point mutation means that there's been a change in one base on the DNA, just a single base. Okay? And so what they're showing you here, here's your DNA template strand on the DNA, and that T that's right there, normally right here, has been changed to an A. And that can, remember when we talked about the proofreading capability of DNA polymerases and when DNA replication takes place? So sometimes DNA polymerases make mistakes. They put the wrong base in, and usually those mistakes get proofread and corrected. But if they don't, then you have a mutation that has been retained on a DNA strand. So if just one base has been altered, that's called a point mutation. There are 2 types of point mutations, though. One is silent and one is missense. And what we see over here is an example of a missense mutation. And so what happens is the change, again, is on DNA originally. That's where the error takes place. And then you move along. You make your messenger RNA. And so you originally had an A there, and that's now a U. When you go on and do translation, you still got your AUG right there. So this amino acid will be methionine. Now, though, you have UCC instead of the original ACC. And if you went out and looked at the codon table, UCC specifies serine. Now, the rest of it hasn't changed because you always move to the next 3, the next 3, the next 3. So you've still got CAC coding for histidine, so forth and so on, until we finish our peptide. Okay, so since we changed in the peptide, the protein, threonine, an amino acid, actually changed to serine, this is called a missense mutation. And sense here is referring to the amino acids. Missense, you have the wrong amino acid that has been put into the protein or the peptide. And remember, it's the gene on the DNA is ultimately coding for a protein. The protein has to be impacted in order for the trait to change. So in this case, this peptide has changed. It's only 5 amino acids in length. You've changed one of the amino acids. Pretty good chance this peptide isn't going to do the job that it's supposed to do. So we call that a missense. A silent change. What if we made a change here in DNA, did transcription, made our messenger RNA, and read the codon table, but no amino acid had changed? Go back and look at your codon table. There's more than one codon, more than one series of 3 bases that specifies each amino acid. So just because you have a point mutation on DNA, it doesn't necessarily mean it's going to change an amino acid in the protein. It depends on what the actual mutation is and where it occurs. So if you have a change on DNA that does not change your sequence of amino acids in your protein, that's called a silent mutation. The reason it's silent is because there's no impact on the traits of the organism. You're still making the same protein. The protein's responsible for the trait. No impact on the actual trait, functions, structure, whatever of the organism. All right, then another type of mutation is a frame shift mutation. And this is a more dramatic type of mutation. And so take a look at what's happened here. So on our original version of the gene, we have TAC-TGGG. Down here you have TAC-GGG. So that TA right there was knocked out. And now when you read your codon table, okay, you've still got your AUG. You got to have an AUG to make a protein in the first place. If that AUG goes away, you're not going to make the protein at all. So AUG is still coding for methionine. But notice up here, originally we had ACC, and that specified threonine. Now we have CCC, and that specifies proline. And on top of that, you know, as the ribosome moves to the next 3, the next 3, the next 3, everything is now thrown off. And that's why this is called a frame shift mutation. Your reading frame is the series of 3 bases that the ribosome is reading at a time. If you knock out an entire base, your series of 3 is going to shift over. So it's going to throw all of the codons off downstream of where that frame shift mutation has occurred. All right, and so if you went through and looked up all of these codons on the translation table, you would see they have all shifted downstream of what we started with. And on top of that, as you went through, if you traced that through and actually wrote out the amino acids, there's no stop codon anymore. We had an UGA stop codon up here, and it's not going to be read that way in this messenger RNA because of the mutation that took place. So your translation is just going to keep going, keep going, keep going until it finds a stop codon or it runs off the end of the messenger RNA, basically. So frame shift mutations can occur. This is an example of a deletion mutation. Your textbook describes those, where we deleted out that T in the DNA right there. You could have an insertion mutation. Sometimes there are extra bases that are inserted in DNA, and those will also throw off the reading frame as well. So just keep in mind, frame shift mutations generally will cause a drastic change in the amino acid sequence of a protein. And now this peptide is so different from the original, it's no doubt not going to do its job. It's not going to be functional. With a missense mutation where you just change a single amino acid in a protein, you really don't know whether that's going to dramatically impact the function of the protein. It really has to be tested in some way. Sometimes missense mutations that cause a single amino acid in a protein to change can have very dramatic effects. Sometimes it can have no effects. Sometimes it might have beneficial effects. It might change the protein so that now it can do a job it wasn't capable of doing before. So sometimes mutations can actually be beneficial to an organism. So in a lot of cases, you really don't know how a mutation will impact an organism until it's actually tested to see what the impacts were. Finally, another type of mutation is a nonsense mutation, and that is where, and sense again here is referring to, what does the codon specify for what amino acid is called for by the codon. Nonsense mutation is where a codon has been changed to a stop codon. So it used to specify for an amino acid in the protein, and now it is specifying for a stop codon. And there's no example of that drawn over here. So you just have to think about what if you had a codon in here and it was changed to UAA or UAG or UGA. Those are your three stop codons. So let's say that happened and you put in your methionine, your threonine, your histidine, but now you had a stop codon. Instead of a codon calling for aspartic acid here, you have a stop codon. You hit a stop codon, translation stops. So in that case, your protein is going to lose whatever amino acids were downstream. They're not going to be there. So your protein's going to be shorter or truncated. And nonsense mutations a lot of times are also dramatic as well. They may dramatically alter the function of a protein. I did want to point out too, missense mutations, I said, you change a single amino acid, there might be a dramatic effect, there might be no effect. A couple of examples of missense mutations where one single amino acid in a protein makes a dramatic difference. Sickle cell anemia is a single amino acid change in a hemoglobin protein. It's due to a single base change on the DNA of those who have sickle cell anemia has resulted in that disease taking place. It's a missense mutation of one of your proteins in hemoglobin. Cystic fibrosis is a single amino acid change, a missense mutation in an ion transport protein that sits in the membranes of your cells, and because of that, it results in those cystic fibrosis patients producing really thick mucus and becoming very susceptible to lots of respiratory infections. So sometimes a single change in an amino acid due to a single base change on DNA can have dramatic impacts on function and even human health. All right, so what causes mutations of genes? Many of them are just spontaneous. So the DNA polymerase enzyme, when it's replicating the DNA of a cell, it makes an error, and that error is not caught by the proofreading mechanisms. It's not corrected, and so then it's retained in the DNA strand, and then it gets passed on to the offspring once it's locked in there. Others can be triggered by things that are called mutagens. So these are things that living organisms get exposed to that make it more likely that a DNA mutation will occur. So because of that, they're said to induce mutation. So radiation, we all know radiation is dangerous. And I know we've talked a little bit before about how there are different types of radiation. Even visible light is a type of radiation. So here we're thinking more about the dangerous high energy types. And ultraviolet light is one. Ultraviolet light can trigger DNA mutations. And then gamma irradiation. That's very high energy types of radiation, and that can damage DNA and trigger mutations as well. In fact, the reason why radiation, high energy types of radiation are dangerous to us is because it damages DNA. It induces mutations, and if you wind up with enough mutations in your DNA, you're not going to be making functional proteins, and your cells are going to die if they don't have their functional proteins. So that's why high energy radiation is dangerous, because it actually alters the DNA in the cells that get exposed to it. And you guys know that, like when you get a sunburn, that's because skin cells were exposed to ultraviolet radiation, and it damaged the DNA and caused those cells to die. And then they peel off, and you've got all those dead skin cells, and that triggers inflammation. So your skin turns red, and due to increased blood flow from the inflammation, and you have swelling due to the infiltration of the fluids and so forth into that area. So you see examples of radiation triggering mutations in your DNA anytime you get a sunburn. Also, we have to be careful about mutagens because cancer is caused by DNA mutations as well. You have mutations in genes that are responsible for keeping the division of your cells under control. And if you have enough of those mutations occur, a cell can lose its ability to control its own division. And so it starts to divide out of control. And if those cells wind up with the ability to spread to other parts of the body, then you have cancer. There are various chemicals that can trigger or induce mutations as well. Some of them just make it more likely the DNA polymerase will make an error during replication, or it might stop DNA replication altogether. There are some chemical compounds out there that can go inside cells and actually react with the bases in DNA and change them. A good example of that is a chemical compound called aflatoxin, which is produced by Aspergillus fungi. That's a common mold out in the environment. And this aflatoxin chemical compound can convert guanine nucleotides to thymine, G's over to T's. So by doing that, it's inducing mutations. There used to be a scare about peanut butter containing aflatoxins years and years ago. And Aspergillus grows in, it's very common out in the environment, and apparently there were problems with Aspergillus growing in big storage bins of peanuts that were used for making peanut butter, and the peanut butter wound up containing aflatoxin. So there was a scare. This is going way back, like when I was in college. There was a scare that if you ate a lot of peanut butter, you might get exposed to aflatoxins, which would make it more likely that you might develop cancer down the road. Just to make you even more paranoid, like you're not paranoid enough, taking a microbiology class. And again, I already mentioned this. Look at your codon table over here. Just because you change one base on the DNA, you change a base on DNA, that change is going to get carried over to the messenger RNA. But then when you do the translation, you make the protein, you got to look at your codon table and see whether anything was actually altered. So for example, if you had a mutation on DNA that caused this UUU on the messenger RNA to get changed to UUA or UUG or UUC, you've still had a mutation on the DNA, a point mutation. I'm sorry, I'm not looking at my codon table correctly here. If this UUU got changed to a UUC, that's still an example of a point mutation, but those two codons both specify phenylalanine. So that's your amino acid in the protein. So your protein's not changing. It's still going to have the same amino acids in it. It's going to be the same protein. It's going to do its job. That's a silent mutation. But if you had this UUU got changed to a UUA, UUA specifies leucine. That's a completely different amino acid. So we're going to call that a missense mutation because we've actually had an amino acid change in our protein. And again, just because you change an amino acid or even a few amino acids in a protein, you don't know what effect that actually has on the protein itself until it's observed or tested. It may have no effect. It could have dramatic effects or anything in between. And as I mentioned, sometimes proteins will change through mutation and now be able to do things they couldn't do before, and that could be beneficial for an organism. So you might have an enzyme that could digest some type of sugar, and now it can digest two types of sugars because its 3D shape has changed a little bit due to amino acid changes. Okay, coming back to our topic of evolution and how organisms change over time, these mutations we've been talking about, those occur all the time. They occur on a regular. There are a certain number of errors that can be expected with about the same frequency, those that don't get corrected. And so because of that, organisms are always accumulating mutations in their DNA. Those just naturally build up over time. And like I mentioned, sometimes those mutations are beneficial to a member of a species, or through recombination events, horizontal gene transfer. Sometimes organisms pick up traits that they did not have before, and those might be beneficial for that organism. So those things are providing diversity and changes in traits. And remember, natural selection as a process, sometimes thought of as survival of the fittest organisms, the fittest members of a species in a particular environmental location. Sometimes those are called niches or niches. They're the ones that are most likely to survive and go on to pass on their DNA to their offspring. And a good example of natural selection, this is what drives evolution, changes in organisms over time. And you think about bacteria that are being exposed to antibiotic drugs in patients. So those drugs are supposed to kill the bacteria, but microorganisms multiply very quickly. There's a lot of DNA replication going on. There are a lot of opportunities for the DNA to mutate or change. That can lead to changes in proteins in the bacteria, and sometimes those changes cause the bacteria to now be able to resist an antibiotic drug that they couldn't resist before. So you think about if you keep taking that antibiotic drug, the resistant bacteria survive, the non-resistant ones die off. And eventually the resistant members of that species begin to dominate the population as they move from one location to another, one host to another. They're spreading around out there in the population. So that's an example of natural selection that has occurred artificially because we've been using these antibiotic drugs for a long period of time. And that is what is driving evolution, changes in organisms over time. Microorganisms multiply fast enough so that in our lifetimes, we can observe the evolution of microorganisms very easily. When you're thinking about large multicellular complex organisms like human beings, you're not going to observe evolution in any major way in a typical human lifespan. Those types of changes have to accumulate over very long periods of time and many generations. All right, another topic I want to mention to you guys is biotechnology. And we're going to talk about that in the next video clip. I realized I had a slide out of place in my PowerPoint because the next video clip, Lecture 11 for Chapter 8, will be on genetic engineering. This is going to be another topic in Chapter 8 where I'm going to hit some highlights. You'll get exposed to more information as part of your reading, but I'm limiting what you're actually going to be required to know for testing purposes in this class. So as always, you want to use your study guide learning objectives for the chapter very carefully to help you zone in on what you need to know when it comes to testing time.